Protein synthesis in 90 seconds. Protein synthesis is the process of making proteins, and it occurs in two main steps, transcription and translation. Our DNA code acts as a recipe to make proteins, and we need to find a way to transport this recipe from the nucleus, where DNA is stored, to the ribosome, where proteins are made. We do this by copying down the recipe in the form of messenger RNA. This process is called transcription and is carried out by an enzyme called RNA polymerase, which unzips the DNA and makes a copy of one of the strands. This copy is then edited in a process called splicing and transported to the ribosome, where it can be translated into a polypeptide. Floating around the ribosome are tRNA. tRNA have a set of 3 nucleotides called an anticodon, and each carry an amino acid. The ribosome uses the mRNA as a guide to sort the tRNA into the correct order. It does this by reading the mRNA 3 nucleotides at a time and attaching a tRNA with a complementary set of nucleotides. By sorting the tRNA into this order, the amino acids they carry are also ordered. The amino acids will then form hydrogen bonds with one another, creating one long string of amino acids called a polypeptide. This polypeptide will go on to make a protein. You can think of it like this. The mRNA is the recipe for a protein. The ribosome is our chef, which uses the recipe to combine the ingredients, the amino acids, in the right quantity and order to make up the final product, the protein. And that's